This video was recorded during a guest lecture that I gave in November 2011 for the Cell and Developmental Biology class at Duke University. The class was comprised of 69 students, mostly biology majors, and the topic was stem cells. I started the class by prompting the students to write a 1-minute paper. So the 1-minute paper, the topic is this: your neighbor's concerned about stem cell research in the US, and what I want you to write about is what does your neighbor already know? And I don't mean your real neighbor, I mean sort of a lay person. What do you think the lay population knows about stem cells in the US? You have a minute and a half to write that out. If you need paper, if you didn't... I gave the students about 2 minutes to write, and then I asked the class for volunteers to share what they had written. Faked? So what Vague said is basically that he thinks that his neighbor is going to know that stem cells come from embryos and not that there aren't stem cells in adult humans, and probably knows that there's some controversy but not why. What else did people say? After the 1-minute paper, I lectured for about 15 minutes defining stem cells and giving some examples from adult tissues. Halfway through the section on hematopoietic stem cells, I set up an experimental problem for the students to work on. Given these two hypotheses and this experiment, this is the experiment we're going to use, what are your predictions? So if the answer is A, hypothesis A, that there are two different stem cells, what would you expect from this experiment? Would 1, 2, 3, 4, a combination of antibody-specific bone marrow cells? Which of those would give rise to a mouse that would live because it has a full complement of blood? Okay, and if it were hypothesis B, then what would you expect? We're going to do something called think, pair, share. So you're going to think about it, you're going to find a partner, you're going to talk about it with your partner, and then we're going to report back to the class. Take a minute to think about it, and then... The think, pair, share activity stimulated conversation. Several students offered alternative explanations that would fit with the hypotheses. Yes, absolutely. So the point, what's your name? Joshua. The point that Joshua just made is, well, couldn't, there's lots of different cells. There's cell type A, cell type B, cell type C, and wouldn't they express lots of different antigens? And so couldn't an antibody bind to cell type B and cell type C? So when I'm isolating... After the discussion, I finished the second half of the lecture covering topics such as the stem cell niche and transit amplification. After the lecture, the students worked in groups to research a stem cell engineering topic on their laptops. But what you're going to do, what your group is going to do, is figure out what is the technique used in each of these engineering cases. How do they go about making stem cells? Both of these are examples where they make stem cells, okay, from cells that weren't stem cells. And then to what end? Why? Why would you do this? What's the clinical application for doing this? And then what are the limitations? So what are... At the end of the class, the student groups reported back to the whole class, and I recorded their responses on the overhead projector. The responses were appropriate, concise, and helped illustrate the medical potential and pitfalls of current stem cell engineering.